this is gonna be one of the, if not the best podcast we have ever done.
Yeah.
Fully naked in a sauna.
Even though that we're not fully naked, and we're not in the sauna, although
Yes.
we are in an unair-conditioned flat in Budapest in August of 2026, so it's pretty much the same.
You know, this will be cut, man. Like, I will cut things out of the things. I just want the energy to, raise the levels. So
Yeah, you gotta establish the chemistry.
Oh yeah, gotta establish the chemistry. You know why? Because one of us, the biochemistry expert. I'm not saying like low level intelligence. I'm saying big time, big level intelligence, like big potato intelligence.
Not sure if I can give any more insight than you could get via ChatGPT, but I'm very honored to have this title
Oh, thank you. But like,
bestowed by you.
yeah. So I love ChatGPT and I use ChatGPT all the time. But sometimes I love people who think and have conversations with people who think. And you're a thinker.
I like to think that I'm more of a doer than a thinker, but
Oh, you're a doer.
coming from you, it must be a high compliment.
Oh yeah. Take it as a compliment, please. Yeah.
Hmm.
So we're gonna take this chemistry to the next level and focus on what has happened, because a lot of things has happened. Spider-Man 4, Spider-Man Brand New Day has come out, and Spider-Man Brand New Day is talking about a shift in Spider-Man's body. This is a spoiler, full of spoilers analysis of what has happened in the movie from a biochemical perspective. You know what? I'm going to ask you questions that may be super easy to answer. I'm going to ask you questions that may be like super sci-fi and you don't even know the answer to them, but let's just think about them as a game or a play, or as if like we can think about like these things easily with ease.
Yeah.
So yeah. So whoopsie, sorry. So I want to tell you what happened in the story, because one of us has seen the new Spider-Man movie, one of us haven't. I have seen the new Spider-Man movie, and the new Spider-Man movie is about a mutating situation. Now, to understand what happened to the beginning, apparently the concept of Spider-Man revolves around a radioactive, some sort of venomous spider biting Peter Parker and turning him into a mutated person who then becomes Spider-Man. I want to ask you two questions. How can a venom make an impact on somebody's body? And how does that venom, if you develop antibodies to this venom, do you have those antibodies forever, or do you have to have repeated exposure to this kind of venom to have antibodies?
Well, it's mixing a lot of concepts in biology in one question. I'm not a toxinologist, like those exist, and I'm not one of them. And didn't really have a chance to learn much about snake venom from biochemistry.
Spider venom.
Or spider venom, much less so. If I had to think about it, I'm not even sure how snake venom or spider venom would be different. But generally speaking, venom contains foreign proteins that shut down one or two key functionalities at a cellular level. So, for example, there is a kind of snake venom that causes a very rapid coagulation of blood. So you literally have all of your blood inside your blood vessels turn solid, and it can be fatal within a couple of hours.
Wow.
There are some animals which are highly venomous that contain neurotoxins that can shut down the ion transport across neurons, that could either make you go completely limp or just turn into a wooden statue-like figure because all your muscles get spasmed.
Crazy.
So venom can act on your body in a number of ways. And yes, there is such a thing called immunity. So in most venoms, there are some kind of proteins which can be recognized as antigens by your body. I believe there are even venoms that would cause damage via an immune flare-up, causing a cytokine storm in your body. That's basically your own immune system's overreaction that makes it dangerous. But yes, they are foreign substances to which the immune system can produce antibodies, but it's not something that's going to happen over a couple of minutes. So with any sort of foreign pathogen, disease, it has to run its course before your immune system starts generating antibodies. Essentially has to learn.
Is that the case?
It's just the time scale that these things go on at a molecular and cellular level.
But you can insert antibodies into your system, and then these can protect you from the venom, right? For example, if you get bit by a venomous spider or a snake, you can get a shot that can protect you.
Yeah, and I suppose most of the time that's just lab-grown antibodies that can help your immune system shut down the action of the venom. So it's basically a shortcut. Doesn't have you don't have to wait for your immune system to start to learn how to generate antibodies and then make them at scale. Because it's a multi-step process. You actually have to develop a certain culture of B cells that generate that specific type of antibody. And that maturation process is not going to happen overnight. This is why most diseases have an incubation period and the time course when you actually develop symptoms. And then some of these you get completely inoculated after first infection, for example, chickenpox. And I would assume this would be similar with a snake or spider venom as well. So if you receive the short anti-venom, then you could, in theory, be more resistant to that particular venom if you were to be bitten again. But then again, I think people say that anti-venom can only be used a couple of times. Yeah, a couple of times until it starts losing effectiveness. So you don't want to get bitten over and over again, even though there is anti-venom available. Not quite sure about how that will work on a molecular level, but that's one of the things.
And the other question part was, once you have these antibodies, and I think you're alluding to this already, once you have these antibodies, how long are they going to stay for? Can they stay forever?
Potentially, yeah.
Okay.
Yeah, so immunity does wane after a few periods, but those cells don't die. Like that culture of cells remains. It just will decrease in number as the infection subsides, and your body is no longer exposed to that pathogen or that foreign antigens. And it's obviously producing those kind of cells and antibodies is going to be less of a priority now. Mm-hmm. And if that occurs on the scale of years or decades, then you could imagine that being exposed suddenly to that particular antigen then will produce a weaker immune response than if you were reinfected 2 or 3 months upon the primary infection. So that would be a significantly different immune response in terms of its strength, but it's not going to be completely wiped out. The reason why we get sick with viruses such as COVID or the influenza virus over and over again, even though we keep generating antibodies and an immune response that most healthy people at least, after each infection is because they keep mutating and mutate at a really fast rate. So the actual antigens that you're going to be exposed to will be significantly different, or different enough that the immunity you have been building up is no longer going to be sufficient to defend you against that viral infection, and you're just going to develop symptoms. I mean, I'm the perfect case study. I've
You
received.
have mutating DNA?
Everybody has got mutating DNA, but
Tell me about it.
I mean, I received 2 shots of the COVID-19 vaccine. I got the mRNA, the Moderna type, and I thought I'm supposed to be reasonably well protected. You know, I was a young man with reasonably good health, but it was right on before the new Omicron variant appeared. And Omicron was significantly different than its predecessor, Delta. So it caused an infection at different parts of the lung, and it was described as a milder variant in terms of its danger to your overall physiology and less lethality, but significantly more infectious. So that's what often happens with these kind of viruses, that they become less lethal but more infectious over time.
Right.
That's sort of an evolutionary
So we
adaptation.
shifted the conversation.
But there is an important lesson here that I didn't get to, which was I got 2 shots. I got the Omicron variant and got really sick.
Okay.
Like for almost 2 weeks, I was
Because you
coughing
lacked the
constantly.
antibodies.
I did not have the sufficient number and the sufficient kind of antibodies to defeat that particular variant of COVID. My mom, who was living with me in a very confined flat, so we had 2 separate rooms, but we shared a lot of spaces, ate together. I mean, she was taking care of me when I was sick, obviously. So she was in an imminent and high risk of getting infected by me, especially with that new more infectious variant. And very interestingly, she did not get sick at all, no symptoms, even though that she had an immense viral load coming from me. And the reason why she didn't was she got 4 shots actually. So she got.
Oh, really?
She got boosted because she was a
Healthcare worker?
frontline healthcare worker. So it was obviously paramount for her to get the latest booster shots and everything. That's why booster shots work, because you get an updated set of antibodies or antigens. Well, in the case of an mRNA vaccine, you get the antigen, or at least a recipe to make the antigen. Your body then makes it, then develops an immune response.
Let's stop there. So it's an mRNA vaccine, which means there's a liquid and there's a bunch of mRNA. What are they, proteins?
So
proteins?
it's an mRNA molecule that's
okay.
packaged into a liposome.
Okay.
So liposome is.
Get that, and then you have the code to create antibodies. Then.
So no, the mRNA is, you can think of it as a set of primary instructions which your cells use to make protein. So it's a template, and it's a template for a certain spike protein of the SARS coronavirus 2.
Right.
Which Is
it true that all viruses have mRNA built in, defined, coded?
No, so viruses, they all reproduce via nucleic acids, just like us, but some work with DNA, some work with RNA.
And what's the difference?
DNA and RNA are very similar molecules chemically. There is one. So DNA stands for deoxyribonucleic acid. RNA just ribonucleic acids, because DNA lacks a hydroxyl group at position 2. So one of the carbons does not have a hydroxyl group attached, and that's thought to be important for long-term stability of the molecule, because hydroxyl groups have a nucleophilic attack capability that can pose a threat to the sugar phosphate backbone. So essentially in an aqueous solution, the molecule can break itself apart via its own chemical reactivity. And so RNA tends to be shorter, more short-lived, single-stranded, but it's the primary source of information by which cells make protein. DNA, you can think of it as a library with a whole bunch of stuff you don't really need. And then your cells
Is there like a
specifically...
famous virus that is using DNA? Because you said some.
Oh yeah,
Yeah.
there are in fact. For example, herpes simplex virus is a DNA virus.
Oh, really?
Yeah, human papilloma virus is a DNA virus, and they can be especially nasty because they actually implant their DNA into your DNA.
That's the thing I want to talk to you about, Adam.
Right.
What is and how does that work? How can you have a mutating DNA? You mentioned you have a mutating DNA.
Everybody's got mutating DNA.
Everybody's got mutating DNA. How can we all have mutating DNA? How does a virus implant DNA into us?
Well, it's a very interesting question. Our genomes are at constant threat of damage. We receive hundreds of...
Is it a threat or a good thing?
It depends. It usually is a bad thing. We usually think of DNA damage as something to be very wary of. Cells definitely are. So our cells are exposed to upwards of 100 DNA insults every day per cell. And those come from a number of different sources. Some are internal, some are external. So there's a reason why people are saying, well, scientists, doctors, who say don't stay in the sun for too long because UV radiation can damage your DNA.
Mm-hmm.
And if DNA damage is not repaired accurately and promptly enough, it can be a source of, either it can lead to either the cells committing suicide or becoming cancerous.
But in your vocabulary, mutating means damaging. Is it true?
No. Mutation in the genetics context refers to a change in the underlying DNA sequence.
Okay.
So, DNA sequence is A's and T's and C's and G's in all kinds of different order. But a point mutation would be, for example, a C changing to a G.
Okay.
Or an A changing to a C, or stuff like that.
Yep.
And those can occur in a random manner for a whole bunch of reasons. For example, or our enzymes are not that accurate, so sometimes they mismatch bases. They don't copy them in the right order. Those are rare, and some of them are dealt with by some repair mechanisms. That are intrinsic to our and most higher organisms. But sometimes even a single nucleotide change can lead to a high risk of cancer, or can be the whole etiology of disease. For example, sickle cell anemia.
Okay.
It's a single base mutation that changes it occurs just the right spot to change a single amino acid in a protein that is just at the right spot. So that particular protein is dysfunctional, and therefore your red blood cells are not as viable as people without that kind of trait.
And I want to circle back to the idea that a virus causing some sort of DNA change. How does that work?
Yes. So viruses can be quite nasty in implanting their DNA into ours, which can itself then become cancerous because obviously a virus has a goal of reproducing in higher and higher amounts.
Can I just ask, how does this work?
Yes. So
I thought we have a DNA, and this can be elongated by a virus's DNA and added together. It's an additive.
No.
Is something?
No, I actually learned about this though. We learned about summer camp. Don't you remember?
Probably.
About transposons.
Okay. Remind me. The audience as well.
So transposons are sections of DNA that can jump across the genome, jump across chromosomes. And some of those are viral origin. So viruses have a very specialized protein machinery that can cut and paste. So they can encounter a section of human DNA. They can cut it, just like do a double strand break. So, and then splicing the first, usually RNA, but just for the sake of imagining it, they splice their own DNA, section of DNA or genome into it. And that's how...
and that causes fever sometimes?
No, fever is a systemic inflammatory reaction that arises due to the immune response.
Okay.
So that's when they successfully, for example, integrated their DNA into human cells. That DNA is then getting transcribed into RNA and then produces the requisite pieces of viral machinery, so new viral particles that get accumulated in the cell, in the host cell. Eventually, that host cell dies, ruptures, and releases all the viral particles. Those viral particles and infected cells then generate a signal for the immune system to start attacking, making antibodies, and generate all kinds of defense mechanisms, of which getting fever is a component of. So it's more due to your immune system than the virus itself.
So back to the story of Spider-Man. So
yeah,
this gentleman called Peter Parker gets bitten by a radioactive super mutant spider, and that spider's DNA then lives on inside his body. Is it true? Is it possible that that spider's DNA is then living on inside of Peter Parker's body somehow?
I don't know of any spider or any kind of organism that would produce a kind of venom that could cause that organism's DNA to get implanted into the DNA of the other organism. A virus can, but then again, the virus wouldn't be, unless that spider is symptomless carrier of the virus, so the virus is not really a danger to the spider itself.
Oh, okay. So that could have been a virus being the messenger of this.
It could be just like the malaria virus.
Like what mosquitoes carry.
Yeah, mosquitoes carry it, or the Zika virus. So they are symptomless carriers of that pathogen that does not really harm them, but through their saliva they inject it directly into our bloodstream.
Right. Okay.
And it harms us.
So the natural conclusion to what I'm seeing right now is that amazing radioactive mutant spider that bit Peter Parker was carrying some sort of spider variant of a virus that led to Peter Parker having the spider variant DNA. And that spider variant DNA inside of Peter Parker's body somehow led to him allowing his body to become more powerful and become Spider-Man.
Yeah, well, this is where we get into the kind of fiction part of it all, because yes, it's entirely possible to have an organism express a kind of protein that you want. We do that in the lab constantly. We use viral vectors that essentially have a piece of DNA engineered by us, and then we feed it to an organism, bacteria, sometimes can even be mice, that implant that particular DNA into the model organism, and the model organism will express that our protein of interest. However, in order to gain all these sort of superpowers, that would require a major reorganization inside Peter Parker's body.
That's right. That's what happened.
That's what happens, but that's not something you could really do externally. That's a developmental process that would really have to begin from the embryonic stage. And
you think so?
it definitely is the case, because it's not enough. It's not just enough to have a cell express a bunch of proteins that you want. Those proteins need to then do the kind of functions that you want. And for example, in order to generate more muscle strength, it's not enough to just make more muscle protein. You have to think about this in a structured way, like how do muscles grow? There's an external stimulus. That stimulus generates a response in the cellular level. Certain genes get activated inside the muscle cells nuclei. Certain proteins get expressed. Those cells communicate with each other, and that sort of cell-to-cell communication, those molecular signaling networks, is something that's intrinsic to the organism itself, and it's a very fine-tuned process. It's not we might get to a point where we understand it fully and able to manipulate it from beginning to end, but it's not going to be via a one-time injection of a substance that would carry instructions for a set of proteins that might be expressed at one particular time. But this is a long process that requires a fine-tuned intervention into the whole co-regulation of the entire system as a whole.
I understand your point, and you're trying to be scientifically accurate, but in the realm of sci-fi, this can work. Because
that's just why it's called fiction.
Yes, and I love it. So what if we let's just accept it for a second. I know we shouldn't, but it's a great thought experiment, I believe, to accept things sometimes. And what if we accept that Peter Parker's Spider-Man's DNA has mutated thanks to this spider biting, and now, due to external stimulus, it is mutating again. Okay? And Peter Parker wants to stop the mutation. And this is a plot actually in the new Spider-Man Brand New Day movie, that he's trying to figure out a way to stop his mutating DNA from mutating to fully becoming more of a spider-like creature rather than, staying human. And this is a fascinating conversation topic, so I wanted to ask you about this because I'm so pumped up about this. So yeah, a matter of fact, I have taken notes in the cinema, and I wanted to read you what they say in the scene that inspired the conversation between somebody called the Hawk, Bruce Banner, who has this green creature living inside of him, and is can basically modify his own body to become the Hawk, and super external, without external skeleton or without anything. It's a mutation of body and becomes a green monster, basically, at the fault of him losing his temper. That's Bruce Banner. And so Bruce Banner becomes a professor at a university in New York City, and apparently he's okay. So it's cool. He managed to figure out some sort of way to prevent the Hawk taking over.
Okay.
And then Peter Parker, Spider-Man, goes to ask the Hawk, Bruce Banner. Is it Bruce Banner? Is it Bruce Banner? Or is it... Yeah, I think it's Bruce Banner, right?
Oh, I think so, yeah.
Yeah. As Bruce to get advice from him. And this is the conversation that ends up happening between the Hawk and Peter Parker. Peter Parker goes up and asks, "You're using target-specific short interfering RNA, right?" I already lost it. I have any clue at all what is short interfering RNA. So
that's actually a very high degree of scientific
thought device.
well, how do you say it?
Expertise.
I wouldn't call it expertise, but awareness for a movie about superheroes.
That's cool. I love that.
The short interfering RNA is a very real thing.
Mm-hmm.
And it's quite fascinating, actually. It's something that arose as a means of defending against transposons.
Mm-hmm.
So over the course of evolution, viruses doing the nasty things they do, they implanted a bunch of foreign DNA into ours. This is why, I mean, it's partly why we usually say 99% of our DNA is junk. It's not really expressed
we're
into
the same
protein.
as a banana.
No, we're not the same as a banana. We are about 55% the same as the banana. I mean, there obviously have to be some sort of commonalities, right? Like there's only one particular. There are only so many sequences that can generate something that's biologically meaningful.
So how does the short interfering RNA come into part?
So the reason why we survive despite having all that foreign junk implanted into our genomes by viruses is because we learn to defend against those stretches of DNA expressing themselves into viral protein. And short interfering RNAs are basically these 20 to 25 nucleotide long stretches of RNA that are anti-sense to those foreign sequences. So, you know how DNA works, it's double-stranded, but there is a specific matching, it matches into, the two strands are matched based on a few rules. So
they have pairs.
They pair. So G pairs with C and A pairs with T. In terms of, in the case of RNA, T are replaced by U's, but then A's pair with U's. So in this particular, it's completely feasible to make double-stranded RNA. It's just not very stable. But that kind of base pairing can serve as a means to shut down the expression of an RNA sequence. So when those foreign DNA segments are then transcribed into messenger RNA to be then made into protein, those short interfering RNAs can get activated. They are the complementary sequences to those foreign RNAs, and they can pair up, and the specific enzymatic machinery then cleaves them into little pieces, and then basically those RNA products get degraded.
Okay, so then Peter continues with this question and says it's like gene silencing.
Yeah, so this is what we do in the lab, because we can engineer short interfering RNAs to suppress the expression of a particular gene that we want. So this really brought about a revolution in genetics in the past for 20 or 30 years, because before that we used, really cumbersome temperature-sensitive mutants that basically the gene gets inactivated off at a particular temperature. They were difficult to work with, or we had to knock the gene out completely, which was very tricky in case of a few genes that were especially lethal when inactivated. Short interfering RNAs don't completely nullify the effect of those genes. They just simply reduce their RNA output. So if those genes are essential for the cell to stay alive, they can be targeted by short interfering RNAs, and you get a very weak and very sickly cell culture, but it's still going to be alive. It's still going to display that particular phenotype that you're interested in.
And so gamma radiation is an external factor that that the hawk is somehow sensitive to. I don't know the exact details, but then Peter asks, so it's like gene silencing, you're incorporating the gamma radiation, and basically, there's no answer to this question, but I assume that the belief is that the external stimulus, the gamma radiation being the external stimulus, somehow incorporated to a device, a wearable watch that Bruce Banner is wearing that produces gamma radiation and is needed for that sort of continuous and relevant external stimulus.
Yeah, well, it's interesting that you brought up gamma radiation, because gamma radiation and basically any kind of radioactivity is highly mutagenic.
Mm-hmm.
It's very genotoxic, in fact. So ionizing radiation produces double-stranded breaks in our DNA, which are considered the most genotoxic lesions. If those are not repaired with high fidelity, they can either cause the cell to die, commit suicide, essentially, or produce all kinds of aberrant translocations, so different sections of DNA then connect to each other, and those can result in fusion oncogenes that basically a promoter section that controls the expression of the adjacent sequence from one part of the genome gets reattached to a gene that you don't want expressed at a high level constantly. And that is how fusion oncogenes arise, and this is what leads to cancer eventually. So this is why ionizing radiation can be so oncogenic.
Okay.
But importantly,
But for talk, it's different.
It's very different than, say, the viral translocations that we discussed previously, because those happen in a very controlled and essentially planned. It's not really a plan; it's a plan made by evolution, I suppose. But it's still a highly controlled and specific process. Gamma radiation is a random process that affects the entirety of the genome in a stochastic manner. So this whole idea that Bruce Banner can, after receiving a high dose of gamma radiation, becomes this green monster is quite, well, I'd just say, improbable.
But it's something that they coded for. So it's like they know that the gamma radiation can be an external stimulus that can enhance the mutation.
But you can't really program gamma radiation to damage particular sections of the genome that you want. Yes, some regions, we used to think that it's all random. We now have evidence that it's not as random as we previously thought, that that's basically all it comes down to is that different sections of the genome, due to their geometry and organization, are more susceptible to damage than others. This does not make gamma radiation an effective tool for modifying the DNA of your target organism. We do that by a bunch of other means that can introduce those double-stranded breaks in a sequence-specific manner.
So maybe you're alluding to something interesting here, that Peter Parker may be going through an even stronger, more realistic mutation than Hawk ever could.
Well, I suppose in a sense that makes more sense, because the spider venom-borne viral particles that we discussed would integrate their DNA into Peter's DNA in a more specific manner than just him getting bombarded with gamma radiation and his all the DNA in his cells fragmenting.
And much like Peter cannot stop asking questions, I'm gonna keep asking you questions.
Please.
So Peter goes on to say to the Hawk, or like Bruce, so you found a way to suppress the expression of DNA, even as it's undergoing the mutation due to gamma radiation, right? Would the same thing be possible with DNA that's mutating for another reason? How would you answer that?
I'm not sure if I understand the question.
So the question is like, let me repeat it. So you found a way to suppress the expression of DNA.
Yes.
Is that possible?
Yes, that's what you do with short interfering RNAs.
Okay. Second part of the question, even as the DNA is undergoing the mutation due to the gamma radiation, right? And then this is like a random question that Peter asks the group.
I think it's mixing two different concepts together.
Mm-hmm.
Yes, gamma radiation and double-strand breaks can introduce point mutations.
Okay.
It's not the main avenue by which they are oncogenic or cause differences in phenotype, but it can happen because when those double-strand breaks are repaired by our cells, we got distinct mechanisms that take care of those. Sometimes certain bases can be lost or mismatched in that process. There is a pathway that's more error-prone in this regard than the other.
What does that mean?
So it basically means that the reattached DNA, so when you close the break and produce a whole chromosome again, won't be quite the same as it was before it sustained that double-strand break.
Okay.
So that can be a source of point mutations, but it's generally rare and it's more of a random process. But it's also due to faulty repair rather than a specific mutagenic process.
Mm-hmm. So the last part of the question is unknown to you, I think, because you don't know the necessary at this point in time the story, what happens to Peter in Spider-Man Brand New Day. Peter has like a shock, a mental shock or something, and this leads to him having like some sort of depression. Like I can literally say that Peter Parker is depressed.
Okay.
Or like very sad, and that leads to him having a hormonal reaction, both from a spider side and spider hormones, and I don't know what spider hormones mean. We can talk about that, and from Peter Parker hormone level, and he wants to eliminate this like part of the problem is that there are too much spider hormones being released in this body somehow, and he develops web shooters, which is crazy. I know it's like mutation of the finest level, but he develops these new web shooters that he didn't have before, and he himself is monitoring these hormones, and he sees like optimal levels for human hormones and too much compared to the previous levels of spider hormones are too much hormones of arachnoid, kind, and so he wants to do some sort of he wants to create a device that can limit the expression of these hormones that are of the spider kind, and so he's asking, would the suppression of the expression of DNA be possible when it's mutating for another reason? That other reason being an unknown reason, or this depression, or some sort of, I don't know, happening in his body. Let me repeat the full question to you in order so you can like think it through.
Sure.
So you found the way to suppress the expression of DNA, even as it's undergoing the mutation due to the gamma radiation, right? Would the same thing be possible with DNA that's mutating for another reason?
So gamma radiation, again, it shatters DNA, breaks it into pieces. That DNA is not going to get expressed. That's not so. That does not. Well,
Then the question, I guess, the question.
yes and no,
is
because as we find out, it's very interesting. It depends on the dose of the radiation. If the radiation is too much, that essentially shatters the whole DNA and breaks it up into tiny chunks. That just expressing that DNA is a geometric impossibility, and that will likely be lethal at that dose for a cell as a whole. At lower doses, when only one or two of those breaks are introduced at a particular chromosome, for example, there are very specific mechanisms that then shut down the expression of that damaged region, and it turns out to be a repair-related and highly necessary mechanism that protects the cell from the accumulation of these truncated or aberrant transcripts, then that can be a source of danger, making all kinds of weird proteins that may damage other proteins inside the cell. So yeah, it's not entirely unfeasible.
Like you can stop the expression of DNA. How do you stop the expression?
There can be many ways. One way is to modify the chromatin architecture. So DNA doesn't just sit naked inside your cell nuclei. It's a chromosome. It's packaged into chromosomes, but chromosomes really only look the way.
For those that don't know, what's a chromosome?
So a chromosome is basically just lengths of a DNA molecule, so like the double helix, but in total there are around 2 meters of DNA inside each of our cells, which are microscopic in size. So there has to be some kind of packaging mechanism in order to contain all that DNA into a tiny nucleus.
The .zip for those computer scientists.
That's a very good analogy, actually, because humans have 23 pairs of chromosomes,
right?
And
okay, I want to know, like, where are these 23 pairs? Is it just a type of thing that we can have? And as each...
It's an evolutionary thing. So it's not necessary for an organism to have 23 pairs. There are organisms with more, there are organisms with less.
Yeah, but...
We just happen to have 23 pairs inside the cell nuclei.
But is it like one chromosome per one cell? Or
no, there are 23 pairs of chromosomes for each cell that has a nucleus.
So now I know that. Yeah. That's cool.
So... and that's 2 meters of DNA in total per cell in every cell that you have in that
for each
has a nucleus.
Or for each cell?
For each cell.
Okay, yeah.
I don't know how long a particular chromosome is. They are different in size and length. So.
Oh, okay, I see.
Yes, and if you just Google chromosomes, like just turn to pictures, you usually see those sort of X-shaped things.
X and Y shape? Are there Y shape too?
Sometimes they are Y-shaped. The center point on that X might be at different points, but usually they are X-shaped because those are... well, let's not get into that. Okay. That's how they look like under the microscope. But they only take up that shape in a very specific point in the cell's life cycle. In the cell's day-to-day life, that DNA is much freer, it's much more unpackaged. Those chromosomes that you would see on Google Pictures are the most condensed, most well-packaged, most compressed or zipped configuration that they can occupy. But at that stage, no gene is getting expressed. So it's geometrically and topologically impossible to really express anything out of that configuration of chromosomes at that point. But that's why but that's not really needed at that point because they take up that configuration when the cells are divided.
So your answer to the question, how can we stop the expression of DNA, is
one way is to condense the DNA into the chromosome architecture into a more condensed state. So just basically as you would compress a file on your computer that's not really readily accessible. And when you need to access the information within it.
There's a police car coming in, and it's like, we have to deal with the police sometimes. Please continue. Thank you, sorry.
They're coming for you.
They're coming for us. We love the police.
So one way to suppress the expression of DNA is to modify the chromatin architecture and make it more condensed. That way, the enzymes that are needed to express that section of DNA to turn it into RNA can't really access the DNA itself as easily, and that's...
Peter Parker's a genius, and he invents a design for a microchip that he somehow, through microneedles, puts on his skin, and this kind of thing stops the expression of this arachnia, this spider-like hormones for him. So how can you make a device? I know it's like very sci-fi question, so it's maybe super improbable and unrealistic to ask you this, but I would love to ask you this because you love asking the right kind of questions, and I love the fact that you're thinking about these questions. So how can you make an external device like a fluid balancing microchip with microneedles, potential that you can put on the skin?
Yeah, I suppose it would be very similar to the kind of blood glucose monitor that you're wearing right now.
Really?
Yeah, I mean, it's completely within the realm of possibility to make a microchip with sensors that could detect a particular molecule in the bloodstream. For example, that could be that particular hormone. Now, hormones are not always proteins.
It's like a pump for insulin as well, but
Yeah, it has a pump that could for example, inject the short interfering RNAs on demand. Now, hormones are not always proteins. Some of them are more simple molecules, but for example, insulin is not a full protein, but it's a peptide that is made using DNA as a set of instructions. So I could imagine making a device that could detect those target protein hormones in the bloodstream, even at very low concentrations, and then on demand, then prompted by that signal, would release short interfering RNAs to suppress their expression further.
This is so cool. So I love how genius you are, Adam, because like you should have told me something that I would have never have thought of or guessed, and now I'm like, huh, I love that. This is Peter Parker. Of course he made this. No problem. Yeah.
Yeah, and hormones are chemical messengers that ultimately result in a change of expression of a particular gene or a set of genes. So
now this is interesting because Peter Parker takes it to the next level, and this is the final question for our conversation. Peter Parker makes a solution for himself, which is he stops overregulating or deregulating. He has the perfect amount of spider hormones to have the, capability of Spider-Man and not be overloaded, sensorily overloaded. And then he asked the question, how can he make the same kind of situation for all kinds of things? That is a multimodal blocker for some sort of talent or some sort of X-Men-like capability. In the movie, there's a person who's a telepath, and that telepathic power is stopped by Peter Parker's new invention when they put this like controller on her, and being a multimodal thing, I want to know like how could some sort of talent or how could some sort of power be detected by a device like that? Is it a phenotype based, search, or is it like a DNA based search? Is it two together? How do you think this could work?
Well, I suppose if those mutants or X-Men have a particular genetic feature in common, then you could detect that using the DNA analytics, very similar to what we use in criminal forensics, for example.
So you think the DNA of X-Men people are alike and they're unique?
If it is, then that would be a way, but I'm not sure if it is. Or
I think that's a good question, but I think the whole premise of the X-Men universe is that everybody is unique.
Yeah,
In that sense, everybody has their own DNA
it's
spectrum.
kind of a contradiction
Yeah,
because they all have different abilities.
Yep.
So they are very different in that regard. And if it all would be genetically controlled, which physically and biologically is questionable, but if you just assume it to be so for the sake of argument, then that would be a highly specific and very, very different genetic composition for every one of them. And it would be quite the opposite of a common genetic theme that would link all of them together as a separate race, so to speak.
Yeah, so one can make the argument that they're all like individual based, but then you would have to make a detection of like
Then you would have to have
individual talent.
you have to have a
And then
whole
how do you stop that
library or
individual talent?
database of all of them with all of their unique features and unique genetic components that are responsible for that. But then you might as well just have all their genomes on file. Just like how a
full conversation
Yeah.
topic, because there is a plot device or like a plot point in this story where this telepathic X-Man has a sister who the government has been experimenting on. They do have data on her sister, but Spider-Man doesn't. Spider-Man is the one that invents this novel technology that blocks all inhibitors. So I pose a question to you again: what if we didn't have a library? How could that be like fast and advanced testing of a certain kind of talent and power that could be stopped by an inhibitor, a genome, or some sort of information device that could tell us more, or is the brain structure?
Well, if those powers are genetic in origin, then just like how we discussed, siRNA would be a type of inhibitor. An hormone could be an inhibitor. Some hormones work by shutting down the expression of certain genes. You could target that particular protein with antibodies. There are a number of ways which you can suppress the expression of particular genes in an organism. Not sure how well those would work in humans, but
Yeah, so the question I think is powerfully strong and super hard to answer, because first you have to detect what's the anomaly. So there's an anomaly relative to the base of a human being. Then
but
there's
it's not that difficult to detect if somebody just
super tall or something. Yeah.
just manages to pick this table up with one hand and swing it across the room.
Yeah, but so detection is like easy then. But then okay, so
Identify the genetic background of that superpower might be more tricky though.
okay, yeah. But why?
Well, because genes don't come with instructions that are readable. They are readily human readable. It's a process called annotation. That's something that we did during the Human Genome Project. And that is partly why siRNAs became that handy, is because previously there were mutant screens, so you just introduce a random mutagenic agent in the organism, and then you see what it would do to the function, what it would do to its phenotype. So mutating a gene might change a phenotype. Then you could infer what the original purpose of that gene was. And with particular genes, this is not as easy. This is why siRNAs, you can silence the expression of some genes, observe the phenotype, and then infer the function of that particular gene.
I think that's a beautiful lesson in this, because like the talent and the superpower of heroes and X-Men are sometimes unknown to the public, because we cannot know it. Where does it originate from, or how does it work? And yet the really person knows exactly how to use them, and that is beautiful.
Yeah, well, I mean, in most cases, even the creators of these movies and comic books are quite agnostic to the underlying mechanism by which the heroes gain their power. I mean, they might describe initial events just like getting irradiated with gamma rays or getting bitten by a spider, but how does that actually transform their cells
We
into
know because we think about this. Yeah.
producing the super phenotypes is something they just leave up to. I guess they leave it an exercise for the reader, right?
Absolutely. Yeah. All right. Thank you so much for the listeners. Let's do a recap what he have talked about. So, do you want to do a recap, or can I do it with you? So first we talked about what did we talk about, Adam?
Are we talking about sources of
The
mutation?
origination, yeah.
I talk about sources of mutation. I hope so, at least, that now you know what the difference between a point mutation and a double strand break is. What could be a source of a double strand break, why they are dangerous, how they might affect the underlying expression of the genes for extra credits, since it's a real and active area of research, but very interesting and important nonetheless. How the expression of genes get modulated to begin with. What do chromosomes and chromatin architecture have to do with that?
And literally we question
How we can interfere in that process with siRNAs, I think that would be an overall summary of what we talked about. As far as the superheroes, I suppose you are more of an expert in them than I am, but
that's cool, man. Yeah, so I love this conversation topics because I think I learned so much from you, and thank you so much for being an awesome conversation partner. And yeah, so here we are on a summer day, just enjoying life and making the most out of our theories.
I mean, sweating our balls off, just trying.
Yeah.
I mean, that's such a vibe, though. I really appreciated it.
Yeah. And we end on a high note. So thank you so much for listening. All right. Bye.